Welcome grade 11s. I want to welcome you to the Junior Tikkie Summer School. So there will be a series of videos which will be live streamed in the next few weeks on our Junior Tikkie.online platform. Please use the given material as revision and also look at all the grade 11 content on our platform. So let's get started. I am Ms. Kutsia, and today we will look at grade 11 life science papers from November 2021. The next two questions we are going to look at is question 2 and question number 3. So they form part of section B, which is a total of 100 marks out of 150. So our first question that we are going to look at is the structure of the nephron. So this is the functional unit of the kidney. So this forms part of the excretion system. So again, a suggestion is to fill in the labels in pencil and very quickly, not to waste time before you forget certain labels. In this question, you will also see that you have been given a table with certain data points in it or results of an experiment that has been done. So this can be or can form part of the investigative question. You have to make sure that you understand what is given in the table for you to be able to answer these questions. Before we read through the question, let's just have a summary and look at what happens and what the function is of the nephron. In the left-hand corner, you will notice that I have placed the complete structure of the nephron. So I'm going to divide it into sections so that you can understand what the functions of each of the parts of the nephron is. So the first section that we're going to look at is known as the Malpighian body. So our first label is a blood vessel known as the afferent arteriole. Then our second one is the efferent arteriole. So what you need to notice or what you need to remember for these two blood vessels, it's very important to indicate that it is the afferent and efferent arteriole. You cannot just use the terms afferent and efferent. So if you look at the picture, you will notice that the afferent is wider than the efferent. So that is in there for a specific reason. So the reason why the afferent is wider is because it's going to allow more blood to move into that blood vessel. The efferent is a bit narrower because it allows less blood to move. The reason for that is our next structure, which is the glomerulus. It looks like a ball of yarn in the middle of the Malpighian body. It's going to create a hydrostatic pressure on the inside of that glomerulus in order for the filtrate to move into Bowman's capsule. So I always tell my learners that Bowman's capsule is the part that looks like Pac-Man that surrounds the glomerulus. So within my Bowman's capsule, the outer layer of Bowman's capsule, it consists out of squamous epithelial cells. The inside of Bowman's capsule consists out of cells known as podocytes. Then the end of my Malpighian body, or just the start of my proximal convoluted tubule, contains cuboidal epithelial cells, which contains microvilli, and that will obviously be for reabsorption of certain vitamins, minerals, and substances. The next section, we will just focus on the glomerulus and the podocytes. So the blood vessel that you will see here is a capillary that forms part of the glomerulus. The podocytes are cells with finger-like projections that literally wraps around the glomerulus. So the glomerulus has small little pores for microfiltration, and as these podocytes wrap around the glomerulus, it forms little gaps or splits for ultrafiltration. Our next section is the proximal convoluted tubule. So this is the section where most of or a bigger variety of substances are reabsorbed. So in this section, we will have the reabsorption of glucose, amino acids. We will have vitamins and minerals that's reabsorbed in the proximal convoluted tubule. The next one is the distal convoluted tubule. So that's the last part of our nephron. So this is where water is reabsorbed and where it is the reabsorption is controlled by ADH, which is a hormone. So let's read through question 2.1. So in the given diagram, they have given you a structure which represents the nephron. So the nephron is the structural and functional unit of the kidney. And this is where reabsorption of various substances take place. So if you have a look, the first question asks identify label A and B, and you will see that that forms part of my Malpighian body. Then name the process that takes place at C. So that will include A and B, that's the entire Malpighian body. So if we look at question 2.1.3, it says the concentration of various substances in the blood, the filtrate, and the urine is given below. So remember, when it's blood, it's still in the afferent and efferent arteriole. Filtrate you will find in Bowman's capsule, and the urine will obviously be in your collecting ducts, pelvis, the pelvis and ureter and bladder. So there they've given you the three locations, so the blood, the filtrate, and the urine. It's very important for you to go through all the values given. So which of the substances shown in the table did not move from the part labeled A to B? So from the glomerulus to Bowman's capsule. So then you just go and have a look at the values that was indicated. So if we look at proteins, for example, you will see that in the blood it was 8, in the filtrate 0, and in the urine 0. Glucose was 10 in the filtrate and in the blood, but 0 in the urine. So that will be your proteins. So then it is present in the filtrate, but was completely reabsorbed in part D. So you go back to the part that says D, that's the proximal convoluted tubule. And you should know the function here because that will be glucose. Then also one of the substances that reaches the highest concentration in urine. So this will be a waste product, and that will be urea. So if you go to your table and indicate the highest value in urine, that will be the answer. Then the next question is 2.1.4. Explain two structural adaptations of part labeled D. So again, part labeled D is the proximal convoluted tubule, and when they ask you to explain, you need to mention the structure and then also explain how it is structurally adapted for that specific function. So let's have a look at question 2.1.5. They say list one way in which the information in the table would differ if it were applicable to a patient suffering from diabetes mellitus before any treatment. So remember that this section is regarding a healthy person and versus someone that has diabetes mellitus. So what would be the difference? So they say list one way, but please note that it was a 2-mark allocation. So let's have a look at our memorandum, 2.1.1. They asked us to identify the parts labeled A and B, which A is the glomerulus, B is Bowman's capsule. Together it forms the Malpighian body. Then name the process that takes place at C, which will then be either ultrafiltration or just filtration as they have indicated in this section. So let's move on to question 2.1.3. So here they say which of the substances shown in the table did not move from part A to B, and that is the protein. So if you look at the table, you will see that in the blood we had 8 grams per 100 cubed centimeters, and in the filtrate and the urine there was 0. Proteins are too large, the molecules are too large to move through the micropores of the glomerulus. So then the substance that is present in the filtrate, but it was completely reabsorbed. So that will be 0 in the urine, but there will be traces in the blood and including the filtrate. So this will be glucose. If we then move to the next one, you will see where it says which of the substances shown in the table reach the highest concentration in urine. So if we go to our table, you will see that it's urea, because here you will see that urea has the highest value for grams per 100 cube centimeters in the urine. So that's excreted from the body. So then they want us to explain, 2.1.4, to explain two structural adaptations for part labeled D. So your proximal convoluted tubule is convoluted. So that means it sort of forms a wave-like structure if you look at the structure itself. So the reason for this is to allow sufficient time for reabsorption. So it slows down the filtrate in the nephron to allow for maximum reabsorption. The next section says that the capillary network is very closely linked or close in contact with the tubules. So this facilitates with reabsorption, so it's faster. Then also the cells that lines the tubule. So this contains a lot of mitochondria for active reabsorption. Please just note they're not active absorption, it's reabsorption. And then the cells of the tubule have microvilli. So these microvilli increases the surface area for maximum reabsorption. Then 2.1.5 lists one way in which the information in the table would differ if it were applicable to a patient suffering from diabetes mellitus before any treatment. So the biggest thing here is that the person with diabetes mellitus will have large amounts of glucose in the urine that's present. Question 2.2 is our first investigation in this question paper number 1. So usually out of my experience, out of previous question papers, 90% of the time they will ask you an investigative question based on your cellular respiration or even photosynthesis. So as a suggestion, when you're writing a question paper, you usually get 10 minutes reading time. My suggestion is to page straight through your question paper towards your investigative question and use that 10 minutes to make sense of your experiment. Because if you do not understand what the experiment is about, you will not be able to answer any of the questions. So before we continue, let's just go through a summary of an investigative question, certain things that we need to know. So, firstly, we will have a look at what is the aim. So the aim of our experiment is very important because you need to know what this experiment is about for you to be able to answer the questions. Then a hypothesis is your suspicion or a proposed explanation based on the limited evidence that you have. So this will be what you think will happen, what the results will be of your investigative question. Then we have 3 variables. So we have the independent variable, the dependent, and a fixed variable, which is also known as the controlled variable. So your independent variable is something that the scientist chose or decided. So that's something he will change, and the changes that's made is what he decided on. Then the dependent variable is something that's measured, and then your fixed variable is something that remains unchanged, so it remains constant throughout your experiment. So your independent variable, if you have been given a table, will be the first column. And this you will also find on your x-axis of a graph. Your dependent variable will be the second column in a table, and this will be found on the y-axis of a graph. And then lastly, your fixed variables will always be mentioned in the description. But please take note that they can ask you to identify fixed variables that's not mentioned. So you have to think a little bit about these fixed variables. I find this one the most difficult. And then it is quite difficult, but most of the time it's mentioned in the description. Then the reliability. So this is how consistently a method is measured. So that means that the consistency throughout your experiment stayed the same. Then the validity. So this is how accurately a method is measured. So that means if you repeat the experiment, it is done exactly the same. So this will usually refer to our fixed variables because our fixed variables will affect the validity of the experiment. So let's read through question 2.2. So firstly, it states, write a hypothesis for this investigative question. So for you to be able to answer this question, you need to know what this experiment is about. Then 2.2.2, they say name the gas that's released during cellular respiration. Then 2.2.3, how was the test for the gas mentioned in question 2.2.2 carried out? So how did I test for this specific gas? Then question 2.2.4, why were germinating seeds used in the experiment? Then 2.2.5, explain why the results was different in the control part as compared to the experiment. So if you look to the right hand side, you have test tube A and test tube B. So test tube A is my experiment where I have germinating seeds. They are alive. They are undergoing cellular respiration. Test tube B has dead seeds. So that means that there's no cellular respiration taking place. 2.2.6, why was a cotton wool plug used in the barrier instead of a rubber blocker? So you'll see there at the top of my test tube, it says I have in test tube A and B both stoppers. So here they made use of cotton wool. Then question 2.2.7, the size of the seeds and the rate of metabolism in the seeds vary, and therefore the results obtained in this experiment may not be reliable. How would you improve the reliability of this experiment? So they have given you a statement and said specifically what was not kept constant throughout this experiment. So you need to explain based on the statement that they've given to you, how will you improve the reliability of the experiment? Question 2.2.8, give a reason for keeping both sets of apparatus in a cardboard box. So basically no light that enters. And then 2.2.9, which two environmental factors must be kept constant during this experiment? So let's move over to the memorandum of question 2.2. So the first question was to write a hypothesis for your investigative question. So remember, you need to know what the experiment was about. And what we were testing is which gas is released during cellular respiration. So here you could have said that the gas that's released is carbon dioxide that's released during cellular respiration, or you could have said oxygen. So this question, there's no right and wrong. It just has to be based on the investigative question or the investigation that we are performing in this question. So you get 2 marks for giving that statement. Then they want you to name the gas that's released during cellular respiration. That's carbon dioxide. And then question 2.2.3, how can we test for this experiment? So you will see if we go back to our experiment with our test tubes, you will see at the bottom of the 2 test tubes, there were clear lime water at the bottom. So as soon as carbon dioxide moves through the clear lime water, it changes into a milky white color. Okay, so question 2.2.4, why are germinating seeds used in the experiment? So the reason for that is that these seeds are actively growing. So therefore the rate of cellular respiration is quite high. So that will make the time that we do on our experiment a bit shorter until we get results. And then also they use more energy than any other part of the plant as they require the energy to actively grow. Then 2.2.5, explain why the results was different in the control part as compared to the experiment. So remember the control was test tube B, which contained dead cells, and then test tube A contained germinating cells. So my control had dead seeds. So that means that there was no cellular respiration that took place because the seeds were dead. So that means there was no carbon dioxide that was produced. So that means also, as a result, the clear lime water would have remained clear. There would not have been a change to a milky white color. Question 2.2.6. Why was a cotton wool plug used as a barrier instead of a rubber plug? So there are spaces between the fibers, so the cotton wool almost acts like a little sponge, so it doesn't block the inside or separate the inside of the test tube from the atmosphere. So that means there is still gases that can move freely through or diffuse through the cotton wool. So that allows the carbon dioxide to move upwards and out of the test tube. Or you could have said that the rubber stopper does not allow the movement of carbon dioxide into and out of the test tube. So that means that there will be no results that's obtained. Then question 2.2.7. The size of the seeds and the rate of metabolism in the seeds varied. So that means it was different. And therefore the results obtained in this experiment was not reliable. So how would you improve the reliability of this experiment? So you can repeat the experiment a few times. You can use a bigger sample of seeds. So instead of using 60 seeds in one test tube, you can use a bit more, 70 or 80. So this will increase your sample size. Then question 2.2.8, give a reason for keeping both sets of apparatus in a cardboard box. So some of the seeds in this container contain chloroplast, and therefore that means that photosynthesis will take place. And this will obviously influence the results, your end results, because in photosynthesis we use carbon dioxide. So that's going to lower my carbon dioxide levels instead of increasing them. Question 2.2.9, which two environmental factors must be kept constant during this experiment? So here it will be temperature and light, as that will affect the rate at which photosynthesis will take place. Not photosynthesis, sorry, cellular respiration. And the light will obviously increase the rate of photosynthesis, lowering my carbon dioxide levels. Our next question is question 2.3. So here they give you an extract explaining how insulin works with blood glucose levels, and then also what it means if someone is insulin resistant. So this is also an important section for you to read through, as it sort of works like a comprehension test, because they can ask questions based on the extract that's given, and then they can also ask questions that you had to study for this specific section. But before we go into detail with the extract, let's just quickly go through our summary again. So let's look at my summary. So my summary will explain the influence of insulin and glucagon on blood glucose levels. So in this extract, we were focusing on insulin, but I'm going to explain to you both sides of the hormones regarding blood glucose levels. So in the middle, we will start with a normal blood glucose level. So as soon as we eat a high carbohydrate meal, so what that means is carbohydrates is sugars, which is broken down to a simple sugar like glucose. So this is if you have a chocolate, a burger, any starchy meals like chips or pastas or bread even, that will increase the sugar or glucose levels in your blood. Then there will be a signal will be sent to the pancreas, specifically to the islands of Langerhans, to secrete insulin. So insulin is a hormone that will then move into the bloodstream, which will then move to the liver or even to your muscles. So this will then stimulate the liver to then convert excess glucose. So meaning glucose that's not used for cellular respiration, and that glucose will then be converted to glycogen, which is then stored in the liver. Then in the muscle cells, it will stimulate the cells to take up glucose, and that glucose will then be used for cellular respiration. That means that the glucose level in the blood will then return, or it will lower down back to normal. So let's say one morning you have forgotten your food at home and you didn't eat at all. So what happens here is that there will be a drop in your blood glucose level. So that drop in the blood glucose level will then send a signal to the islands of Langerhans in the pancreas. This will then release a hormone called glucagon. So glucagon will then enter the bloodstream and then travel to the liver. So this will then stimulate the liver to then convert glycogen, which is the stored form of glucose, back into glucose. The glucose will then be released into the bloodstream. Then there will be an increase in the glucose level in the blood, which will then return the blood glucose level back to normal. So question 2.3, let's quickly read through the extract. So in the extract, they say that insulin is a hormone that allows cells to absorb and use glucose, which is a simple form of a carbohydrate. So it regulates the amount of glucose that circulates the bloodstream. So this must be maintained at a normal level. It also instructs the liver to store excess glucose. So that is something you need to remember. It's excess glucose, not just glucose, the extra glucose that's not used for cellular respiration. So in people with insulin resistance, so meaning people they produce or secrete insulin, but the insulin, they are resistant to it. The body doesn't react to the insulin. So in people with insulin resistance, the cells are unable to use insulin efficiently. So when the cells cannot absorb glucose, the levels of glucose in the blood will build up. So therefore the pancreas will actually make extra insulin to overcome the low rate of glucose that's absorbed. And diabetes is less likely to develop. So this means that the blood glucose level will stay within a healthy range because of the excess insulin that's secreted. So over time, the pancreas' ability to then release insulin actually starts to decrease, which leads to a development of a type 2 diabetes. So in question 2.3.1, they say name the gland that secretes the insulin. So the gland, not the cell, the gland. 2.3.2, why is glucose required in the cell? So what is the purpose? Why is glucose important? Then question 2.3.3, describe how insulin resistance leads to a type 2 diabetes, and it's explained in the extract. Then question 2.3.4, this you have to read very carefully. Explain how a normal glucose level are maintained. So normal glucose level when a healthy non-diabetic person consumes food containing a high percentage of glucose. So it's a normal person, not someone that's insulin resistant or someone that has diabetes type 1 or type 2. So let's have a look at my memorandum. So question 2.3.1 asked us what gland secreted the insulin. So yeah, it is pancreas. I actually do not agree with them for saying that it's the islands of Langerhans, because that is the cells responsible for it, and they specifically asked for the gland. Then question 2.3.2, they asked us why is glucose necessary? What is the importance thereof? So it's our main source of energy. So glucose gets broken down for cellular respiration. Then question 2.3.3, they asked us, how does it work when someone is insulin resistant? So when someone is insulin resistant, so do you see that you don't get a mark for that because they have mentioned that in the question, the body cells becomes incapable of using insulin. So they cannot use it effectively. So they are unable to absorb glucose. So this increases the amount of glucose in the blood, and then it will stimulate the pancreas to secrete extra, even more insulin. So this will then allow the cells to absorb sufficient amount of glucose, but over a long period of time, the pancreas loses its ability to secrete extra insulin. So that will then develop into a type 2 diabetes. And this entire paragraph was given in the extract, so you just had to copy it. Then question 2.3.4, they asked you, how does the body maintain normal blood glucose levels in a healthy person that has a high carbohydrate meal or that had excess intake of glucose? So the glucose levels in the blood will increase. You get a mark for that. Then the pancreas is stimulated to secrete insulin into the bloodstream. The insulin then travels through the bloodstream to the liver. It stimulates the liver to convert extra or store extra glucose in the form of glycogen, and then the blood glucose level will decrease and return to normal. Our last question for question 2 is question 2.4. So this is another investigative question. So this is a bit unfortunate because I know that there are a lot of learners that finds this very difficult, but the notes that we actually work through with these experiments have a lot of similar questions that you can follow. There are some other question papers available on the internet as well that you can work through to see how they ask these investigative questions. So question 2.4, before we just move on, I just quickly want to show you an example of an experiment that I did actually for my grade 8 natural sciences. So I just want you to watch the video so that you can see the procedure that we did, how to test for starch and the presence of sunlight, or if sunlight is necessary for photosynthesis. I'm going to do an experiment to determine if sunlight is needed for photosynthesis by testing for the presence of starch. In front of me, you will see a variation of apparatus that we are going to need for the experiment. So firstly, we have ethanol, which is also known as alcohol. We have a tong. We have iodine solution. We have 2 test tubes already filled with ethanol. Then I have a Bunsen burner, which is an energy source for heat. I have a beaker with water and a leaf in it. So we have 2 leaves, which we will boil in the water for about 5 to 10 minutes. 1 leaf was placed in a cupboard for 24 hours, and the other leaf was actually freshly plucked the morning that was exposed to sunlight for quite a while. So it takes quite a while to put the leaves and wait for it to boil in the water. So what I did over here was I just held on to the beaker while I was stirring the leaves with a glass rod, as the beaker actually gets quite hot very quickly due to the Bunsen burner and the flame that's at the bottom. So again, you just leave the water there. You leave it to boil, and the reason why we boil the leaves in the water is because we want to break up the cell walls, very similar to when we are boiling vegetables, because it actually softens the vegetables, or in this case the leaf. So after the 10 minutes or 5 to 10 minutes, we'll just give it a quick stir, and then we will remove the 2 leaves. Now we will take the tong to remove the leaf from the water. So this will be the leaf in the cupboard. We will place it in the test tube filled with ethanol. I just pushed it down a little bit with a glass rod, and then I did exactly the same with the leaf that was found in the sunlight. So what I'm doing now is that I'm placing the test tube filled with ethanol in the water, and to allow the ethanol to boil. The reason for this is because alcohol, which is ethanol, is extremely flammable. So that means if I place it over a direct flame, it can set alight very easily. So it's for a safety precaution measure. The reason why I'm boiling the leaf in the ethanol is to wash out the chlorophyll. So again, I leave it for about 5 minutes to boil in the ethanol. After the 5 minutes, we will remove the test tube again with a tong, as it is extremely hot. Then you will notice the green color of the ethanol. You will also see the picture in the left-hand corner, where it then shows you a clear and closer look on the color of the ethanol. The reason why it is green is because it contains the chlorophyll. The whole purpose of boiling the leaf in the ethanol was to wash out the chlorophyll. So now the next struggle was to remove the leaf from the test tube, as I noticed my tongue was way too big to remove it. Luckily, the glass rod helped a lot. So I removed the leaf from the test tube and I then added it to a white plate. So the reason why I use white, it just makes it nice and clear for us to see if there is a color change. If starch is present in the leaf, that's either in the sunlight or in the cupboard, it will turn blue-black. You are not allowed to write navy or purple. The color is blue-black or black-blue. So you will see in the animation in the left-hand corner, I'm adding iodine solution. And as I add the iodine solution, it will turn and change into the blue-black color. If the leaf in the cupboard, if I add the iodine solution, you will notice that there's no color change and the leaf actually takes up the color of the iodine solution, which is a brownish color. So therefore, my conclusion in this experiment is that sunlight is definitely needed for photosynthesis to take place. So let's read through my investigative question. So they say that an investigative question or investigation, was conducted to determine whether light is necessary for photosynthesis. Then the investigation was conducted as follows. So we had a potted plant. So we had a potted geranium plant that was kept in a dark room for 2 to 3 days. So the reason why we do is because we do this is because we de-starch the plant. So as the plant is kept in this dark room, it uses the starch stores for the form of energy. So photosynthesis will not take place in these 2 to 3 days. Then we will take one leaf, which we will use to test starch. Then the potted geranium plant was placed in the sunlight for about 4 to 5 hours. Then we take another leaf that was partially covered with a cardboard strip as shown in the diagram below. Usually they make use of foil as well. So anything that blocks out light on a certain part of the leaf can be used. And then we test for the presence of starch of the leaf that was used secondly. Then question 2.4.1, they say, why was the plant placed in a dark cupboard, which I actually already explained. 2.4.2, why was the starch test conducted after 3 days? Then 2.4.3, name the chemical used in testing for the presence of starch. Then question 2.4.4, why is it only part of the leaf that was covered with the cardboard box? And then question 2.4.5, draw a labeled diagram of the leaf to show the results after the test, the starch test was done at the end of the investigation. Question 2.5 is separate from photosynthesis, so this has nothing to do with photosynthesis. Here you had to explain any 3 structural requirements of an efficient gas exchange system. So remember, when you have to explain, you mention the 3 structural requirements and why they make it possible for efficient gas exchange. So let's discuss our memorandum. So on our left-hand side, you will have the questions, and then also in the middle, we'll look at the memo. So question 2.4.1 asked, why was the plant placed in a dark cupboard? So the reason for this is to de-starch the plant. So remember, if we immediately test the leaf, there will be starch present. So we will place the plant into the cupboard for 2 to 3 days to force the plant to use the starch stores, because we want to test that light is necessary for photosynthesis. If we test the plant immediately, we cannot prove that the starch was there because of the presence of light. Then question 2.4.2, why was the starch test conducted after 3 days? So this was to check whether the leaf has been completely de-starched, to confirm that there's no starch. So we just wanted to check that there's no starch possible before we cover the leaf with our cardboard strip. Question 2.4.3 asked what chemical was used to test for for the presence of starch. So this is iodine solution. You are not allowed to write iodine. It has to be iodine solution for you to be rewarded the mark. Then question 2.4.4. Why is only part of the leaf covered with the cardboard box? So here you will see they say to prevent light from falling on the part that has been covered. So that means it blocks out sunlight on that specific section. So this serves as a control and not your experiment, because I'm removing the factor that I'm testing, which is light. Then question 2.4.5, you had to draw a labeled diagram to show the results. So if we look at the diagram, you will see we have a leaf. It does not have to be perfect. The part that was covered with the cardboard box, there will be no color change. It will turn into the brownish color of the iodine solution. So there's no starch. And then the two sections that was not covered turned blue-black, which contains starch. So then you get 1 mark for stating which part changes color and which part does not, and then also for the correct diagram and shading. Question 2.5, that's not part of photosynthesis. You had to explain 3 structural requirements of an efficient gas exchange system. So remember again, you mentioned 3 structures and then why they make it efficient for gas exchange. So question number 2.5. They say the surface area of the gas exchange organ must be large. So the reason for this is to allow for maximum diffusion of gases like oxygen and carbon dioxide into and out of the bloodstream. Then the surface area must be moist because my gases must be in a solution form, so they dissolve and diffuse into the moisture. The surface area should be thin. So this allows for rapid diffusion of the gases. Then I need a transport system. So my blood system must be available to transport these gases from one point in the body to another. Then there should be adequate ventilation. So this should ensure ease or to ensure that oxygen moves into and out of the lungs. And then lastly, the gas exchange area should be protected. So this is usually by means of the pleura or the rib cage, so that it allows, it says because the gas exchange system is very thin and delicate, so it has to be protected, especially protected from drying out as well. Thank you, Junior Tukkies. Please make sure that you watch our next videos.